So today, what I'm going to walk you through is a series of studies that we've been doing in my lab in Boston to try to understand how we can use our innate immune system to control anti-tumor immune responses. I'm very eager to get the feedback from the community on this topic, so feel free to ask as many questions as you can towards the end. Now, generally speaking, the work that's done in my lab is focused on a very simple question, and that is, how do our cells distinguish each other from the microbial cells that we may encounter? Typically speaking, we think of our own cells, such as this cell in the center of your screen, as having a healthy or symbiotic relationship with other cells that share the same genomes, like dendritic cells and hepatocytes, for example. And we think about our own cells having an antagonistic relationship with the microbial cells that may enter our normally germ-free tissues of our bodies. But we know that not all interactions with our own cells are productive. And in fact, in the case of cellular transformation, we know that transformed cells can ultimately be as dangerous to the host as an infectious agent, leading to the giant set of diseases that are, of course, of major concern today, known as cancer. Now, typically speaking, the fields of immunity to infectious disease and the field of immunity to cancer are considered separate fields of study, meaning that if you go to an international conference on infectious agents, you do not typically hear scientists talking about cancer, and the opposite is also true. But I would like to argue, and I think many in the scientific community agree with this point, that the lessons that we learn from both the left and the right sides of our screen are informative for the other. And the reason for that is because cancers and infectious agents share several common properties. If they are not controlled by our immune system, they will both multiply, they will spread, and they potentially can cause the death of the host. And it's the job of our immune system, either naturally or therapeutically, to prevent the multiplication and the spread of these threats to our own viability. And that's what we're going to be discussing today. Now, anytime you're talking about detection of threats, which is what we're going to be discussing today, we need to go back to basic principles. And the basic principle about how our immune system detects any threat that we face is basically described through the actions of dendritic cells. Dendritic cells represent the apex regulators of our immune system. These cells, either directly or indirectly, control the actions of all other immune cells in our bodies, including the targets of very successful immunotherapies, such as T cells. And as early as 1989, the concepts by which dendritic cells operate in this fashion were illustrated, at least holistically, by the late and great Charles Janeway Jr., who introduced the idea that for the last 50 years, there has been a dirty little secret of immunology. And that secret was that we generally would discuss that antigens, cancer antigens or infectious disease antigens, will be needed to stimulate adaptive immunity. And that's true. The secret, though, would be that antigens alone are rarely immunogenic. The immunogenicity of an antigen is revealed when you mix these antigens with adjuvants. Adjuvants were therefore needed for antigenicity, and this was the original dirty little secret that founded the modern field of molecular innate immunity. The outcome of this is the concept that the innate immune system, which was activated by these so-called adjuvants, was necessary to weaponize your adaptive immune system to hunt down and eradicate the antigens, meaning that the fields of innate immunity and adaptive immunity were not separate entities. The innate immune system was capable of capturing antigens, presenting these antigens to T cells, but also providing the signals to those T cells that were needed so that you could generate robust protective immune responses. Those signals were first provided in the context of these experimental tricks that we would use, or chemicals called adjuvants. But these adjuvants ultimately were representative of a larger biological principle. Adjuvants are chemical mimics of infectious agents. I'd like to argue that these days, there's a new dirty little secret of immunology. The new dirty little secret of immunology can be illustrated here, that necessity equals sufficiency. What do I mean by this? Well, as we know, we do live in a field today where it's very easy to do genetic analysis. And there have been numerous genes that have been knocked out in the immune system. And that once knocked out, there's a defect in immunity or there's an enhancement of immunity. And because of that, there's been a tremendous explosion of biological interest in coupling genes that you could knock out that are required for immunity to therapeutic pathways where you target those gene products. The general belief in the field is that cellular activities that are necessary for immunity are sufficient for immunity. And because of that, there's been a lot of work to identify protective antigens in the context of cancer or infection, or CD40 agonistic antibodies, the use of recombinant interleukin 12 or interleukin 23, or the use of recombinant interferon as an immunotherapy. The justification for this is exactly what I mentioned. Because antigens are needed, because CD40 signaling is needed to stimulate immunity, therefore all you need to do is provide antigens or provide CD40 agonism, and you can actually result in immune stimulation. The dirty little secret is that most of the time, adjuvants or therapeutics that stimulate a single innate immune activity rarely induces therapeutic immunity. Agonistic CD40 antibodies, recombinant IL-12, and antigens on their own do not provide durable immunity, which is the expectation of the public. And so the question is, why is it that we know of all these pathways that are necessary, but none of them are sufficient to trigger durable immunity? What I'd like to argue is the reason for this is because our immune system is not designed to operate with a one signal paradigm. Our immune system is designed to operate in a multi-signal paradigm. And so we've introduced the idea recently that there are 5 dendritic cell activities that are required to generate durable T cell activation. These activities should be familiar to many in this room. Antigen presentation, co-stimulatory molecule upregulation, the production of effector cytokines such as IL-12, but also in addition to this, you need a very strong ability to migrate to the lymph node from the site of the cancer or the site of infection or the site of immunization, because it is there within the lymph node that dendritic cells can talk to and interact with the antigen-specific T cells that they are so desperate to find. And in addition to this, work that's been done in recent years has helped illustrate the importance of inflammasome-dependent interleukin-1 beta as a key driver of memory T cell responses. So in the absence of IL-1 production, you can generate effector T cells, but memory T cell production is greatly diminished. So what this paradigm would suggest is that any therapeutic approach that targets a single entity or even multiple entities of this pathway without hitting all 5 would result in an incomplete, durable immune response. The ideal scenario to best weaponize our immune system is to trigger all of these pathways. How would you go about doing this? One way is to go back to the classic view of Charlie Janeway, which is that toll-like receptor agonists are fantastic agents of adaptive immunity. And the reason for that is because toll-like receptors, which are activated by infections and infection-like molecules like LPS and RA48, derivatives of which are in the clinic today, these chemicals will signal through tolls and activate 3 of the 5 signals I described to you. Antigen presentation on MHC, co-stimulatory molecule production, and effector cytokine generation. And when dendritic cells are exposed to these chemicals and they are agonized through the toll pathways, we consider these cells to be activated. And that's the term that many in the world use to define a stimulated dendritic cell, an activated cell. But we also know that toll-like receptors in dendritic cells are woefully incapable of driving strong amounts of IL-1 production, and they're also poorly capable of inducing dendritic cell migration. I'm going to show you evidence for this in the next few slides. And what we found over the recent years is that if you combine signals of infection with signals of tissue injury, which we call hyperactivators. You can add to the activities of a dendritic cell the ability to migrate to the lymph nodes so you can increase the probability of finding cognate T cells, and you can add the ability to stimulate the inflammasome to drive interleukin-1 production through the actions of the famous NLRP3 protein that drives this activity. Under these conditions, what you can see is that if you stimulated dendritic cells with Toll ligands alone, you would engage 3 of the 5 critical signals needed to generate adaptive immunity, durable adaptive immunity. But if you combine infection signals with these so-called hyperactivators, you can add all 5. And the reason why we call these hyperactivators is because they are adding signals on top of an already activated cell, and therefore we call these cells that have been stimulated with both infection signals and injury signals hyperactive dendritic cells, as opposed to active dendritic cells. One of the classic chemicals that we use to mimic infectious disease, mimic tissue injury, is this hyperactivator known as PGPC. It's a lipid, an oxidized lipid that I'll describe now. So anytime that any of our cells die, they could be through physical damage or a programmed death pathway, the cells will always release chemicals into the extracellular space. Some of these chemicals can be immunosuppressive, some of them can be inert, but one of them is inflammatory, and that is this oxidized chemical known as PGPC. And what we found over the years is that PGPC, when combined with toll-like receptor ligands, results in these 5 activities of a dendritic cell. And therefore, we consider LPS, for example, or TLR ligand plus PGP-treated cells to be hyperactive dendritic cells. So what does this look like? So we can look visually at mouse bone marrow-derived dendritic cells, and we can ask visually, what do these cells appear as? These are naive dendritic cells. Green, we have stained the plasma membrane. Blue is the nucleus. You can see these cells adopt the classic amoeboid shaped structure. If you stimulate these cells with LPS, not much changes. The cells still have this amoeboid shape. If you stimulate cells with the classic adjuvant that's most commonly used in the United States, aluminum hydroxide, or in combination with the toll-like receptor ligand LPS, these cells are known to undergo pyroptosis. They will die, they round up, and most clearly they do not look like dendritic cells. If you treat cells with these hyperactivating chemicals, LPS and PGPC, the cells adopt the classic dendritic cell morphology. They become extremely elongated, so large that it's hard to find them in the same field of view without cutting off their extensions. These morphologies are classically associated with an enhancement in cell migration, and we were also interested in this because when we did RNA sequencing analysis of differentiated dendritic cells, again from mice, in culture, you can see unique transcriptional programs that are induced by either LPS or LPS plus PGPC or some of these other agonists that I described, with the most notable transcriptional signature that was uniquely induced by LPS and PGPC being one associated with cell migration. These cells were upregulating many chemokine receptors, including CCR7, that results in dendritic cell migration in vivo. And for that reason, we wanted to know, do PGPC-treated cells exhibit enhanced migratory activity in an animal? And so what we did is take dendritic cells ex vivo and stimulate them with any number of these chemicals. And then we adoptively transferred these dendritic cells into the skin of a mouse, effectively doing a vaccination of the mouse with the skin, with the dendritic cells itself. And then asked 24 hours later, how many of those dendritic cells migrated to the lymph node? What you can see is that PGPC plus LPS treated dendritic cells exhibited a massive ability to migrate very high levels to the lymph node as compared to toll ligand stimulated dendritic cells alone, or alum, which was almost completely incapable of driving dendritic cell migration. This slide was important because it proved 2 points. One, that toll ligands are very poor, as we knew, at inducing dendritic cell migration. And as I mentioned, if you do not migrate, you cannot effectively stimulate the T cells you need to do. but also that hyperactivators add to the activities of LPS to allow these dendritic cells to migrate to high numbers to the lymph node. Which dendritic cell state is most effective at inducing or T cells that eliminate tumors? That's what we're going to talk about for the next several slides. We took a very simple approach, a cancer vaccine approach. We can take antigens from a tumor, and again, I'm illustrating a human, but all of this is in mouse studies. We mix these antigens with chemical agonists that would represent hyperactivators or other stimulators of dendritic cells, and then we inject these chemicals into the mouse and then ask, can these chemicals protect against cancer? We're going to start with the B16 ova melanoma model. This is classically used by many in the community, although I'll show you a few slides later where we used other tumors. We implant these mice with these cancers and let them grow to a tumor size of about 4 millimeters over about a week. After the tumors have formed, we give these mice our cancer vaccine, which includes LPS and PGP-C as our hyperactivator, as well as antigens collected from tumor cell lysates. But we've used a variety of different antigen sources. As long as the antigen came from the tumor, you'll get the same type of results I'm going to show you. We do a few boosts, and then we track what happens with the cancer itself. And what you can see is that if you do not give these mice any immunotherapy, in the black line, these mice will sadly die in a few weeks. If you give these mice cancer antigens plus a pyroptotic-inducing adjuvant, LPS plus alum, commonly used on the market today, you get almost no benefit. But if you take the same antigens and you mix them with our hyperactivators, we get durable immunity for several months. This slide was important to us because it illustrated that antigen identification alone is not sufficient to confer protective immunity. If you have the correct antigens in complex with the wrong adjuvant, you either have no protection at all or very minimal protection. But if you take those antigens and you mix them with the proper adjuvant, you get durable immunity. So why are these mice so protected? Well, if we look 200 days after the tumor has been eliminated and we ask what's going on in the body of the mouse where the tumor once was, you can see you have a very high percentage of CD8 positive T-resident memory cells, TRM cells, that are effectively surrounding the place where the tumor used to be. TRM cells, as you're going to hear much more about throughout this Immunoschool, are considered some of the most potent effectors of the adaptive immune system to control tumors. And we believe that the reason why these vaccines are working so well is because we're creating TRM cells that do not go back to where the immunization was. Instead, they go to where the tumor was and eliminate the tumor and then guard that tumor from ever recurring again. We're excited about this finding because we can take T-resident memory cells or circulating memory cells out of these survivor mice, meaning that mice that 200 days before eradicated the tumor. And we can ask, do these CD8 T cells kill the same tumor ex vivo? The answer is yes. What we're measuring here is cell death. You can see that these T cells will kill B16 ova tumors, but they'll also kill B16 tumors that had similar antigens that we used in our immunization. And this antigen-specific killing is indeed antigen-specific because these T cells have no ability to kill a different tumor cell. And perhaps most importantly, we can take these CD8 T cells and then transfer them into a naive mouse. And you can see if we transfer both the resident memory and the circulating memory cells into these mice, we get 100% protection from tumor challenge. These mice that I'm showing you on the right were never immunized. They were only challenged with the tumor and had these T cells transferred into them. The contribution of resident memory in bold and circulating memory in dashed lines is about 50% each, but the combination of them both provides maximal immunity. And that means then that our hyperactivating cancer vaccines are enlisting CD8 T cells that can hunt down and eradicate tumors independent of the actions of other immune cells, or at least that's what we believe. And perhaps what is most useful, especially in this context of today's world, where we're all talking about how many COVID boosters we need to receive, it would be very nice if we could start creating vaccines that would indeed last for a long time. And so we asked, how long do our immunizations work? In this experiment, we simply immunized mice with ova-albumin as an antigen at 8 weeks of age, and then we waited over a year. We immunized them with one boost of ova and then challenged these mice with B16 tumors that expressed ova. And what you can see is that if we used LPS plus PGPC as our vaccine adjuvant early on in life, we get 100% protective immunity. No other adjuvant provided this durable immunity. And if we knock out NLRP3, the inflammasome, within these mice when we immunize them, we lose almost all the protection that we saw. This allows us to conclude then that inflammasomes are important to allow cancer vaccines to last into old age. So at this point, I'm going to shift now to the question about old age. What I've shown you so far are studies where we have immunized mice young in life and then studied them young in life. Or we immunized mice young in life and then waited until they became older. But we also know that cancer is largely a disease of the elderly. Sadly, most of us know someone who has had this problem, and that problem manifested later in life. Now, there have been many defects in immunity that have been documented in the elderly. The primary immune response is diminished in the elderly. The recall response is diminished in the elderly, and there are many other aspects of immune system development and change that occur as we get older. It is not news to document immune defects with age. In fact, there are too many to consider. The problem here is not counting the number of problems that you have in the immune system as we get older. It is correcting those problems. And that's what we're going to talk about for the next few slides. So one of the things that mouse models do is provide a very nice representative of what happens in humans. So in human beings, we see exactly the same results that I'm going to show you here in mice. And again, this presentation is all in murine cells. As we get older, as mice get older, as they go from 8 weeks to 90 weeks of age, you can see that the total pool of CD8 T cells is diminishing. And perhaps what is more alarming as far as vaccines go, if you look on the right side of your screen, if we look at CD8 T cells, you can see that as we get older, the naive pool of CD8 T cells dramatically drops. As we get older, we're losing the naive pool of CD8 T cells, and that would be expected to dramatically diminish the ability of vaccines to work because you simply don't have many naive cells to be charged by a vaccine. This observation has also been made by many others in humans. What we've also found, which also represents the studies of others, is that dendritic cell migration is dramatically diminished with age. If you look on the left side here in mice, you can see that at 8 weeks old, dendritic cells will migrate at a decent amount, not great, from the skin to the draining lymph node. If we use LPS plus PGPC, as I showed you earlier, we get a greater enhancement of young mice migrating their dendritic cells to the lymph node. But with old mice, 68-week-old mice, you can see in these black bars almost no migration of these dendritic cells. And what we've been pleased to find is that PGPC can correct this age-associated defect almost to young mouse levels. I'd like you to keep that in mind, because what I'm showing you here is the first evidence that we can indeed correct an age-associated defect, at least in the innate immune system, the ability of dendritic cells to migrate to the lymph node. And when we've done experiments using human monocyte-derived dendritic cells from donors that were over 70 years of age, and this is our human studies here, you can see that if we look at an in vitro dendritic cell migration assay, LPS plus PGPC-treated dendritic cells migrate at a far higher frequency than any other dendritic cell that we've examined. So, with less CD8 T cells and less migratory dendritic cells in an elderly mouse, what happens to cancer and cancer therapies as we get older? We started this experiment by simply asking, let's just give these mice cancer. So if we implant these mice with B16 ova tumors and we compare 8-week-old mice to 20-week-old mice, you get about exactly the same kill curve. These are non-treated mice. They're only just given cancer. As you go older and older, 8-week-old to 40-week-old, 8-week-old to 68-week-old, or 8-week-old to 92-week-old, you can see that the difference spreads. The older the mouse is, the faster the mouse dies from the same exact tumor, meaning that intrinsically, as mice get older, they're losing their ability to protect themselves from these cancers, even in the absence of an immunotherapy. So now let's give these mice an immunotherapy. Let's give these mice PD-1 based checkpoint inhibitors, although we've done this with other checkpoint inhibitors as well. And what you can see, and we were surprised to find, is that while young mice are very nicely protected from these growing tumors if you give them PD-1 treatments after the tumor has formed, elderly mice have almost completely lost responsiveness to this checkpoint inhibitor. And this to us was very interesting because it's the same genotype of mouse and it's the same genotype of tumor. And it's the same immunotherapy. The only difference here is the age of the mouse. Yet the mouse loses responsiveness. But perhaps what was interesting to us is that regardless of age, if we gave these mice the same immunization regimen I described to you with dendritic cell hyperactivators plus tumor cell antigens, you can protect these mice at 8 weeks or 68 weeks of age. We can look and see, is a hyperactivating vaccine special in this context? And the answer is yes. Only when we use tumor antigens plus hyperactivators in red, but not when we use LPS as an adjuvant or LPS plus alum can we protect elderly mice. And here are the tumor growth curves, just in case you're interested. You can see PGPC treated mice, very strong protection from tumor growth. All the other ones grow at very high rates that ultimately result in lethality. And we've seen similar results in these models as well as in the CT26 model, which is utilized in BALB/C mice as opposed to black 6 mice. So this is not unique to the cancer, and it's not unique to the mouse strain. And this is where it started getting interesting for us. Because CD8 T cells were contracting with age, we were intrigued that our vaccines, our hyperactivated vaccines, would be effective. The reason for this is because when we do hyperactivating vaccine immunizations in young mice, you can see that if you deplete CD8 T cells in black, we lose the ability of dendritic cell hyperactivators to protect these mice. CD8 T cells are required to protect young mice from cancer. But if we do these same experiments using dendritic cell vaccines, using hyperactivating adjuvants in old mice, CD8 T cell depletions in red do not diminish effectiveness of the vaccine, but CD4 T cells are now required. This phenotype was striking, and it would really illustrate some change in the way that our immune system is operating with age. So let's look more clearly at CD4 T cells in elderly mice. What we found is that if we look in the tumor microenvironment of immunized mice, look at the tumor infiltrating lymphocytes, you can see that an unimmunized old mouse, there's a lot of regulatory T cells in there. This is well characterized data. If we immunize these mice with LPS and PGPC plus tumor antigens, we diminish the frequency of Tregs, we enhance the frequency of Th1, meaning interferon gamma producing T cells, or at least the ratio of gamma producing T cells to IL-10 producing T cells, meaning that we are warming up or inflaming the tumor microenvironment. And that prompted us to collaborate with the Broad Institute here in Boston to ask, what do the T cells look like at different stages of life? We did single-cell RNA-seq analysis of the lymph nodes that were draining the site of immunization of these mice. We decided to focus on the lymph nodes here because of the complexity of the tumor microenvironment. These tumors are growing at different ages, at different rates. It's very difficult to normalize and draw any conclusions. But the healthy draining lymph node from the skin, distal from the tumor itself, provided us with a consistent material that we can start comparing age. What was nice to see is that regardless of age, we can identify all the classic T cells that we want to by single cell analysis. And we can see that these T cells are very different. 8-week-old T cells in blue look very different than 90-week-old T cells in orange. And if we compare mice at different ages that were immunized with the same vaccine, LPS plus PGPC plus OVA as your antigen, you can see that you have very different types of T cells. So what are the differences? Here we're comparing mice that were immunized at 8 weeks of age or 90 weeks of age with LPS, PGPC, and OVA. 8-week-old dendritic cells will be enriched, or excuse me, 8-week-old T cells will be enriched on the left, 90-week-old transcriptional signatures are enriched on the right. Lots of genes that come up. We're going to walk you through some of these modules. One of the module is as expected. I already described to you that naive T cells are diminishing with age. We can see that very clearly on the top left of your screen. As we get older, we lose many of the signatures associated within T cells of a naive repertoire, and we start increasing just intrinsic T cell exhaustion in the lymph nodes of old mice. But what was perhaps most interesting is after immunization, you can see that old CD4 T cells had a higher frequency of Th1 expression, including interferon gamma. And what was most interesting to us was the cytolytic score. These are transcriptional signatures that are classically associated with cytolytic activity associated with CD8 T cells, but we see them in CD4 T cells with age. This signature was represented at the level of the granzymes, interferon gamma, as well as perforin. And this prompted us to ask, is it possible that our old CD4 T cells displayed cytolytic activity? So we immunized mice, and then we isolated CD4 T cells or CD8 T cells from older young mice and asked, can they kill or degranulate ex vivo? Here I'm showing you CD8 T cells that were isolated from mice that were immunized at 8 weeks of age or 92 weeks of age. You can see if we look at degranulation, you can see that CD8 T cells retain some ability to degranulate if we immunize them with hyperactivators, even as the mice got older. But this degranulation was really the only marker that we saw for function. These T cells were unable to kill B16 cells as assessed by flow cytometry to monitor the frequency of live T cell cancer cells in this culture. 8-week-old or mice immunized at 8 weeks old had CD8 T cells that rapidly killed within 15 minutes the B16 cancers. 92-week-old T cells could not kill these cancers. If we look at LDH release, which is again is an independent sign of cell death, you can see that old T cells cannot cause cancer cell death. Young CD8 T cells can do so. So as mice are getting older, their CD8 T cells are losing the ability to operate as cytolytic agents. And interesting, CD4 T cells are gaining that ability. If we look at degranulation of CD4 T cells when co-cultured with cancer cells, only old T cells had this ability to degranulate. Only old T cells had the ability to kill B16 ova cancers, and only old T cells had the ability to actually lyse these cancers. Meaning that with PGPC, LPS, or hyperactivating immunizations, CD4 T cells, specifically in elderly mice, were gaining cytolytic activity. And this activity in vitro correlated with activity in vivo. We can take CD4 T cells out of an old mouse that was protected from cancer and transfer these CD4 T cells into a naive mouse and protect this mouse from cancer. CD8 T cells were much less effective. B cells were ineffective. Monocytes were completely ineffective. The strong protective immunity when transferring to a naive mouse was unique to CD4s, and perhaps most importantly, the recipients were depleted of CD8 T cells, meaning that these CD4 T cells that were transferred into the mice were able to eliminate the tumors without any contribution of resident CD8 T cell activities. So what does all this data mean to us? It allows us to go back to basic principles and ask, why has it been that cancer vaccines have been so ineffective in the past? And they have not been completely ineffective, I should say. It's just that they have not been game-changing for the therapeutic ambitions of those who undertook them. If you look at activated dendritic cells, such as those stimulated with poly-IC, so stimulated with CpG, they are capable of eliciting some, but not all of the 5 dendritic cell activities I described. Alum, or the ASO chemicals from GlaxoSmithKline that are so commonly used in the clinic today, are inducing dendritic cell death. And of course, a dead dendritic cell can do very little. We would argue that based on our knowledge today, we would have expected that these approaches would not yield complete protective immunity as we would have hoped. In contrast, when we think about hyperactivated dendritic cells, they do exhibit all of the activities I described, but the therapeutic potential of these cells is completely undefined. And so what I'd like to close this presentation with is an idea, a model, and that is that hyperactive dendritic cells, because of their ability to migrate at high levels, add memory signals, as well as the classic first 3 signals that we all taught about in graduate school and medical school, they're capable of most effectively eliciting cytolytic and interferon-producing T cell responses. In the young, these activities come from CD8 T cells. And perhaps most interestingly, in the elderly, these activities come from CD4 T cells. And when we think about a disease as nebulous and as dynamic as cancer, it may very well be that hyperactive dendritic cells, meaning innate immune control of dendritic cell activities, would provide the flexibility that we need to confer anti-tumor immunity at any stage of life. And so with that, I'd like to close by thanking the scientists who did this work. This was largely the work of Danya Savaki, a fantastically talented, world-class scientist who I've worked with for many years. She had some terrific help from Stephanie Kennedy in my lab, as well as many collaborators around the world. And of course, I'd like to thank you all for your attention. I'll be glad to take any questions or feedback that you may have.